So hello students, how all of you doing great. This is Basavaraj Sir, your biology master students. Welcome to Vedantu NEET English. Now, students, this is a part 2 of the microbes chapter. If you are in class 12th, this chapter is very important, but it is actually a very easy chapter. Now, let's say generally speaking, in this chapter, the most difficult part, or let's say the conceptual part, is the sewage treatment plant. Now, there are many students who try to learn it, they try to mug it up, but they do not remember it at the end of the day. Students, in this class, you will be able to understand the entire process of your STP in the best way possible, students, in the best way possible with the help of amazing slides, students. So, students, if I'm audible and if I'm visible to all of you, quickly smash that like button right now, students. Smash the like button and let's start the class, students. Let's start the class, all of you. Yes, if I'm audible and visible, students. If I'm audible and visible, quickly add any emoji in the chat to let me know I can start the class, students. Again, my classes are very detailed. My classes are very, technical. So if you have any doubt anywhere, quickly drop a comment in the chat and I will reply to all of you, students. But please focus in the class, nothing to be distracted about. Okay, students, shall we start, all of you? Students, shall we start the class? Yes, ready all of you, students? Ready all of you? Yes, ready? Good evening all of you, students. Good evening. Now, I will teach you very simple manner. Just follow my guidance and you will secure the highest of marks. At the end of this class, students, at the end of this class, I will be doing a lot of questions. So hopefully stay till the very end if you want to solve all those questions, because I have around 25 questions for all of you, 25 to 30 questions for all of you today. Now, if you want to solve even more questions, in my Telegram, Basavaraj Sir Biology, I have already added a DPP there. So please go check the DPPs. It's a free DPP for all of you. It's a practice paper. Now, students, now the first thing which I want all of you to understand is, please watch the first part, right? Please watch the first part. This one includes all the starting part of the chapter. And students, here are the amazing comments. So you know, I always acknowledge the comments. Comments are always positive. It drives teachers to teach more. Yes or no? So students, after this video, quickly drop a comment, all of you. Now, students, now let's understand how do we use sewage. How do you treat sewage? Now, first of all, students, tell me in the chat right now, all of you in the city or in a village. Students, tell me in the chat, where are you from? All, tell me, where are you from? Let's have the very basic question. Where are all of you from, students? What is your current location? I will not track you down or anything. So tell me, students, what is your current location right now? All of you. If you're from a city, or if you're from a small town, or if you're from a village, doesn't matter anything, students. Where are you from? Where are you watching this lecture right now from, students? Quickly in the chat, students. Now let's say in a small town or in a big city, right? Either it is a small town or it is a big city, usually sewage is released. But have you ever wondered what is the end of this entire waste? Where does the waste actually go? It can't disappear, no, students. Obviously the waste cannot disappear like that, like magic. It can't disappear. The waste has to go somewhere, right? So generally, students, this entire waste is removed into a natural water body. So all the waste from the town, all the waste from the city is sent to a water body, students, sent to a water body. Now, students, tell me, all of you, can we directly send the waste? Let's say waste is coming from town, waste is coming from city. Can we directly send it to a natural water body without any treatment? All of you, students, tell me. All of you should know the meaning of sewage. Students, the meaning of sewage is the waste, the human excreta, everything, right? All of this sewage has the organic waste, microbes, lot of pathogenic bacteria, human excreta. Now, students, imagine if you send all that waste directly. If you send all that waste directly into a natural water body, will there be issues? So tell me, will there be issues? Because if we send all the waste directly to natural body, untreated sewage into a natural body, will there be issues? Will there be problems? Will there be consequences? Yes, obviously. Imagine you send it directly. What will happen? What will happen, students? What will happen? Right? What is the problem here? Right? water animal starts dying because all the waste is sent, making water unfit for use, spread of waterborne diseases. Yes or no? Imagine at the end of the lake, at the end of the natural water body, there is a small village. That small village is depending on this water for their requirements. Now, if you send all the waste directly, that village is harmed. Yes or no? So, can you send waste directly into a water body? No, it is not possible, students. It is not possible. It is not possible to send the waste untreated. So, what is the solution here, students? The solution is very simple, students. What we should do is we have to add something called as STP. STP is also called as sewage treatment plant. Now, once you add a sewage treatment plant, all the waste from here, all the waste from here is collected into a separate compartment called as sewage treatment plant, and everything becomes proper now, students. Now, what is waterborne disease? Waterborne disease are the disease which are spreading through water. Cholera, right? Cholera. If you drink contaminated water, you can get disease. They say, right? Don't drink water, students. If you're from here in India, all of you know, you might have seen that unhygienic pani puri person. He will be scratching everywhere. Same pani puri he'll put in the hand, will give you, right? In that water, there can be a lot of microbes, right? If you eat the same dirty pani puri, gone. Students, please avoid street food. I'm telling you, right? So those are waterborne diseases. So what we need to do is we need to treat the water. We need to purify the water. That is called as STP. Sewage treatment plant. The whole need of STP is to purify the water before leaving it into a natural water body. So with that basics in mind, shall we start, students? Shall we start, all of you? Students, this right here is a STP. This right here is a STP right now. Students, do you see? It is very large. बहुत बड़ा है. Like it's very large. Very large. Can you see? It has multiple components everywhere. One component here, one component here, one component here. Now, after there are multiple steps here, it is sent to the water body. Okay, it is sent to the water body. Okay, students. Pani puri, you can tell pani puri. I know what is water ball. We're telling water ball. Tell pani puri. No, it's fine. I'll understand. Okay, students. So this is the entire sewage treatment plant. It is very large. Entire thing is sent here, students. Now, shall we start step by step, students? Very simple. The intro is set. The pace is set. Shall we start the class now, students? Can I get some fire emoji, students? Can I get some fire emoji in the chat, all of you? And all of you, quickly smash that like button, right, students? Quickly, all of you, smash the like button, and we will start the entire STP right now. The best way possible. Best way possible from my abilities, I'll teach you right now. Shall we, students? Shall we start, all of you? Students, shall we start? Yes? Okay. Now, students. Now, I want all of you to understand, in STP, right, sewage treatment plant, in STP, there are 4 different chambers here. Students, how many? There are 4 different chambers. Chamber 1, chamber 2, chamber 3, chamber 4, and chamber 5. 4 different chambers. In every single chamber, step by step, the water gets purified. Very simple, very simple. Now let's understand what exactly is happening in every single chamber now, students. Every single chamber. Now we will start with the first one, students. First one. Shall we start? Okay. Now before we start the first chamber, you need to realize this entire chambers is divided into 2 categories. The first category is called as physical removal of waste. In the first chamber and the second chamber, in the first chamber and the second chamber, there is something called as physical removal of waste. And 3, 4 and 5, all 3 are chemical, right? We use chemicals here, or in this case, apart from chemicals, we use actually microbes here. We use microbes, students. We use microbes here. Clear, all of you? Now, in physical removal, students, physical removal is also called as what? Primary sewage treatment. So, in examination, if they tell you physical removal, Is it primary or secondary, students? Tell me in the chat quickly. Physical removal is primary or secondary? Students, quickly in the chat. Physical removal of waste is called as primary sewage treatment, where physical removal of particle happens through filtration and sedimentation. Through filtration and sedimentation. So primary is physical removal. Perfect, students. Perfect. Now, here I have mentioned again, students. Here I have mentioned again first and second chamber. First and second chamber, both of them are primary sewage treatment. So the meaning of primary sewage is what, students? The meaning of primary sewage is what? All of you? It means physical removal of waste. Physical removal of waste. Clear all of you, students? Notes I'll get I'll upload in Telegram. Okay, Basavaraj Sir biology. Now, students. Now, let's understand the first chamber, students. Let's understand the first chamber. This is the first chamber here. First chamber. Now, in first chamber, there is a technique called as sequential filtration. Now, students, I want all of you to tell me, students, I want all of you to tell me 5 waste items you throw on daily basis. All of you go. Students, 5 items which you throw on daily basis. Let's say you throw a bottle, you throw one more shoe. Let's say you threw a shirt. Dustbin. Done. Now, in this sequential filtration, you might observe, students, here. Here we can see the pore size is big here. Big. As we go from here to here, do all of you notice the size of the pore is decreasing, and the substances which are being stuck here, the waste which has been stuck here, is getting smaller and smaller and smaller. Here we can see large items as the grid. These are called as grids. As the grids are becoming smaller and smaller, the particles left behind are smaller and smaller. Yes or no, students? Yes or no, all of you? Yes or no, students? This is called as what, students? This is called as what? Sequential filtration. By sequence, we are filtering the substances from larger particles at the far right corner. As we move from here to here, we get the small grid size. In the small grid size, almost all particles are being removed now. Students, any doubt in the first topic? Any doubt in the first chamber, students? First chamber. In the first chamber, we do something called as sequential filtration. Here we remove floating solid debris. Large particles are left behind. As we go from here to here, smaller particles are left behind here. Students, any doubt here? First chamber, all of you. First chamber, any doubt, all of you? Students, first chamber, any doubt right now? First chamber, where we do sequential filtration. In the first chamber, we do the sequential filtration. Solid particles are removed. Solid physical property waste is removed here. Okay, students. First one is done. Now, that is called as filtration tank. Okay, that is also called as filtration tank, because we did filtration here. Filtration we did here. Filtration tank. Done, all of you? Done. Filtration tank, sequential filtration we do here. Okay. Now, students, second tank. Second tank is called as sedimentation tank. Do all of you know what is sedimentation? Now, you might have did this experiment in chemistry. We take a glass of water. In that water, we mix sand. Now, we completely stir. We take a glass, we add mud, soil in it, and we completely mix it. Now, have you noticed after some amount of time, the sand will undergo sedimentation because of gravity? Students, how many of you observed that? Raise your hands. Students, how many of you have observed that? Raise your hands, students. Raise your hands. All of you. How many of you observed that? That is called as sedimentation. Sedimentation of sand. That is, water will be separated, sand will be separated because of gravity. Because of gravity, water and sand are separated there. Okay. Same concept, students. Same concept here. Here what we do is, we make sure all the waste is stagnate here. In this one chamber, we keep all the waste. Now, because of gravity, what happens? Because of gravity, all the grids, pebbles, everything will start to go down. Everything will start to move downwards. And The bottom part is called as primary sludge. This primary sludge is again sent back, students. Primary sludge is again sent back. Yes, that is called as, yes, Luna. That is called, Akshita, is called sedimentation. Right? Now, because of gravity, everything on the bottom is called as primary sludge. This primary sludge is again sent back for the filtration tank to purify the water more. Now, whatever is present on the top is called as effluent. The superheat is called effluent. This effluent is sent to the next chamber. Next chamber, students. Next chamber. Okay. Settling down of all small floating particle in the solid debris method is called as sedimentation. Primary sludge, all settled down is the primary sludge. Whatever is present on the top is called as supernatant floating debris. Okay, students. Super debris, students. Students, sound check. Okay. Students, I'll talk to Shreyas, sir. Don't worry. We'll talk to Shreyas, sir. Okay. My students. Clear? All of you clear here? Clear, students, here? I will talk to Shreyas, sir. Don't worry, students. Comments, dekhna chahiye, student. You have to see. Okay. So that is called sedimentation tank. Clear, all of you? Sedimentation tank. Now, students. So we have two tanks done right now. First is called as filtration tank, where we did the sequential filtration. Then sedimentation tank, where we did sedimentation. Now, students, whatever is present on the top, the effluent on the top is sent to the next chamber. Students, first two chamber done. All of you done first two chambers? All of you. The first two chambers are done. The primary treatment is done here, students. The primary treatment is done here. Now we go to the secondary treatment. In secondary treatment, we use what? We use microbes. Students, we use microbes here. Okay. So, students, removal of organic matter from the sewage using microbial action. Now we use microbes right now, students. We use microbes right now. Okay. Now, that is the third one. That is the third one. Now, third tank is called as what, students? Third tank is called as aeration tank. Now, why do we call it as aeration tank, students? Why do we call it aeration tank? Because here in aeration tank, we have a bacteria which requires air. Students, what do we call bacteria which requires air? All of you, anyone who can tell me in the chat? Can anyone in the chat tell me what do you call a bacteria which requires oxygen? Aerobic bacteria. So here in this aeration tank, we use air and we use agitators to spin it around so that more and more bacteria can grow here. More and more bacteria can grow, students. More and more bacteria can grow here, students. More and more bacteria can grow here. Okay. So here we can see, students, we can see the bacteria here now. So we can see organic matter, that is the waste, students, waste here. Students, I took a class today. Rakshita, I took a class today, right? I took Plant Kingdom, I took there. Tomorrow I will take this thing tomorrow there. Tomorrow I will take, not tomorrow, Sunday I'll take part 2. Okay. Students, here in this entire aeration tank, we can see the organic matter, we can see the bacteria here, and we can see fungal filaments here. Now this bacteria and fungal filament, they will join and they'll make a mesh here. Complete mesh. They'll make a mesh here, students. They'll make a mesh. Okay. So mechanical agitators and the pumping of the air increase the number of aerobic bacteria. And what happens, students? They make a mesh. And the mesh is called as what? Flock. Flock. So what is flock, students? Flock is a mesh-like structure, mass of bacteria associated with fungal filaments. So students, technically speaking, flock is nothing but bacteria, that is aerobic bacteria plus fungi. That is called as flock formation. So in aeration tank, we have flock formation. Now students, this flock will eat all the organic matter, all the waste, all the fecal matter, entire waste is eaten up by the flock here. So microbial flocks consume the major part of organic matter. See, all this organic matter will disappear now. Disappear, student, disappear. Now, if you want to understand how does it disappear, you need to understand a small concept called as BOD. Students, shall we start BOD, all of you? How many of you know what is BOD? Can anyone tell me what is the full form of BOD, students? What is the full form of BOD? Biochemical oxygen demand. Now, I will teach you the best way to understand BOD. Ready, all of you? Ready? See, Microbes consume the organic matter, decrease the level of BOD. Okay, let me just teach you one simple way, student. Okay, what is the meaning of BOD? The meaning of BOD is that the amount of oxygen consumed by the bacteria, biochemical oxygen demand, how much of oxygen is required by the bacteria for oxidation of all organic matter in 1 liter of water. In 1 liter of water, how much of oxygen is required for the bacteria to eat all the organic matter? That is for the BOD. Now, students, let me teach you something. Let's say I have one container here. I have one more container here. Okay. Let's say this container and this container, both of them have some amount of liquid in it, students, liquid in it. This container is less polluted. This container is heavily polluted. Okay. Now let's say here we have less polluted water. Here we have more polluted water. Tell me, this is A, this is B. Tell me, students, in which one do you think we will have more organic matter? Where do you think we'll have more waste? Where do you think we'll have more waste, students? In A, do you think we have more waste where pollution is less? Or in B, do you think we have more organic matter? Students, tell me, where do we have more number of waste material? Where do we have more organic matter? Students, obviously here, if it is more polluted, there is more organic matter here. Obviously, there is more organic matter here. Yes. Here we have less organic matter. Less organic matter, students, here. Less organic matter here. Okay. Now, students, tell me, in which one do you think we will have more bacteria, all of you? Tell me, students, which one do you think we will have more bacteria? See, here less food, here more food. Students, you will tell me, whenever someone is giving one free food for you and there is no food, where will you go? If you are a bacteria, where will you go, students? Will you go here or will you go here? Where do you think we'll have more bacteria? B or A? Obviously here, students, here we will have more bacteria. More bacteria here. Here we will have less bacteria. Less bacteria here, students. Less bacteria here. So more bacteria, less bacteria, students. Okay. Now, if more bacteria is here, where do you think is more oxygen demand? Now, these are aerobic bacteria. These are aerobic bacteria. Yes, aerobic bacteria. So, where do you think we will have more O2 demand? Obviously here. Here we have more oxygen demand. Here we have less oxygen demand. Now, students, tell me, if this is a 1 liter water, this also is 1 liter water. Now, if students, where do you think we will have high BOD? Where do you think we will have more biochemical oxygen demand? That is the amount of oxygen required by the bacteria to consume all the organic matter in 1 liter of water. Obviously here we will have more BOD. Here we will have less BOD. Students, tell me, if water is very pure, will the BOD be high or low? If the water is very pure water, will the BOD be high or low? Simple question to all of you. It's a NEET PYQ. It is a NEET PYQ, students. It is a NEET PYQ, all of you. If I will give you pure water, will the BOD be high or low? Very simple question. Pure water I am giving you. Pure water. If I am giving you pure water, BOD will be less. If I am giving you polluted water, highly polluted water if I am giving you, that BOD will be high. So, BOD is very high for more polluted water. BOD is very low in pure water. Students, is that concept clear to all of you? All of you. Is that concept clear to all of you? The concept of BOD. Clear, students? Clear, all of you? The concept of BOD. So, if the water is pure, less BOD. Water is very dirty, very polluted, more BOD, students. More BOD. Students, So let's say I give you this water right now. Let's say I give you this water right now, very high purity water. Now this has to undergo something called as STP, sewage treatment plant. Okay, it has to undergo STP right now. It has to undergo STP. So what do you think our goal is? Our ultimate goal of STP is what? To decrease the BOD, because BOD is very high here. So the ultimate goal of STP is what, students? The ultimate goal of STP is to decrease the BOD, students. All of you. The ultimate goal of STP is what? The ultimate goal of STP. Goal of STP is to decrease the BOD. If you decrease the BOD, obviously the water will become pure. Obviously the water will become pure. Yes or no? If you decrease the BOD. So that is the whole concept of your sewage treatment plant. Sewage treatment plant will always aim at to decrease the BOD. What is BOD? The amount of oxygen consumed by the bacteria for oxidation of all organic matter in 1 liter water. So students, if you see a graph here, oxygen consumed and time. So as we run STP, right? Rakshita, correct. Right. As we run more and more STP, where STP goes on first chamber, second chamber, third chamber, fourth chamber, the BOD will keep on decreasing. Okay. Biochemical oxygen demand tests the measures of rate of uptake of oxygen. Okay. So students, greater the BOD, higher the BOD in the wastewater, higher is the amount of organic matter, greater is the polluting potential. Sewage water is treated until BOD is reduced. So we have to make sure the end product of sewage treatment plant will have very low BOD. If there is low BOD, water is pure. Okay. So sewage treatment plant is treated until BOD is reduced, because whenever BOD is reduced, we get pure water. Why pure water, students? Pure water has less organic matter, less bacteria. Very simple as it gets. BOD is reduced. Done, students? Done. Now, that is called as aeration tank, students. Aeration tank, done. In aeration tank, we try to decrease the BOD. How? By increasing the microbes. Increasing the microbes, and here also we have a flock formation. This entire flock of microbes and fungal filaments will decrease the BOD by eating the organic matter. They're nice, they're eating the organic matter is reduced here. Okay, that is aeration tank. Then students, any doubt till aeration tank, students? Any doubt till aeration tank? The whole point of aeration tank is to decrease BOD. How? By increasing the microbes, by giving them more oxygen, by forming a flock here, and ultimately decreasing the organic matter, and the BOD decreases. Now students, now that is it. Now students, after aeration tank, what do we have, students? After aeration tank, the next tank is called as settling tank. Students, settling tank. Settling tank is just like sedimentation tank. Here also we leave the tank just to settle. Again, because of gravity, what happens? Again, because of gravity, all of this bacteria will be able to go down. So the flock containing heterotrophic bacteria and fungal filaments settle at the bottom. So all of these bacteria, students, all of these bacteria will go down now, students, go down. That is called as activated sludge. Activated sludge is called as activated. Why? Because it has microbes. In primary sludge, there were no microbes, but in activated sludge, we have microbes. Can you see, students? The entire sludge here, bottom. So all the fungal filaments, all the bacteria is settling down. That is called as activated sludge. Now top part is called as what? The clear, this one, the top part is called as clear supernatant treated effluent here. Now this will go to the next, this will be sent to the water body directly. This will be sent to water body because it is clear already. It is clear. Now what happens to the activated sludge, students? Understand. Some amount of activated sludge is sent back here. You might ask me, sir, why? Students, do you remember how is curd made, students? Students, tell me, how do we make curd? Students, how do we make curd? We take milk, fresh milk. In that milk, we add a little bit of curd. The curd has what? The curd has lactobacillus, also called as inoculum or starter culture. We take the inoculum and we add it to the milk. The milk will become now curd. Now from the same curd, we take a little bit of curd to one more milk we add. Yes or no? The same thing is happening here. Here we have bacteria. To make sure in aeration tank we have bacteria. To make sure we have enough of bacteria here. Enough of bacteria here. From here we send a little bit of activated sludge. Activated sludge is sent back here. Activated sludge is sent back here, students. See here. A small part of activated sludge is pumped back into the aeration tank to serve as inoculum. Now, why, students? Why? Because in settling tank, organic matter is reduced. Yes or no? Is reduced. Organic matter is reduced here, and all the bacteria is hungry now. Like all the bacteria is very hungry now, because everything is over now. All the organic matter is over in settling tank. It is over now. Yes or no? It is settling tank, organic matter is over. So all of this bacteria is very hungry. Now where is the food? The food is here. So we send all the hungry bacteria again back to aeration tank. So all the hungry bacteria is going back to the aeration tank right now. Back to the aeration tank. Hungry bacteria. Aeration tank, students. Now remaining part, students, remaining part is sent to the last chamber. Top part is sent here. Can you see the top part is sent here? Okay. Now, what is the last tank called, students? Last tank is called as anaerobic sludge digester tank. It is called as anaerobic. It means what? There is no oxygen here. If there is no oxygen here, can this bacteria which we sent from here, all of this aerobic bacteria, can it survive here? All of you, can all the aerobic bacteria survive here? Obviously not. They have to die now. So all of the aerobic bacteria will die. It will die. Students, do you realize we use the bacteria and we kill them? We use the bacteria to decrease the BOD. We use the bacteria to eat the organic matter. Once they are used, what do? We kill them. Students, are we greedy? No? Crazy greedy we are, right? We will use them until they have a purpose. After their use is over, we kill them. So this is the sad reality. This is the sad reality which we do in the STP. Sad reality which we do in STP, students. Sad reality. What to do? What to do, students? Sad reality. Now, let's say, students, let's say all the aerobic bacteria is dead now. Now, what do we have here? We have all the anaerobic bacteria here. We have the anaerobic bacteria here, students. Now, do we have to treat them? Students, do we have to treat this bacteria? No. This bacteria, let's say this bacteria, if you release in water, it will automatically die, because in water we have oxygen. Yes or no? So students, we don't have to kill them here. Aerobic bacteria will die on its own eventually. But until it is alive, until this anaerobic bacteria is alive, it will use the dead body of the aerobic bacteria and digest it and release methane, CO2 and hydrogen sulfide. See here, anaerobic bacteria digest the activated sludge, the dead bodies. Activated sludge release gas like methane, carbon dioxide and hydrogen sulfide. That is also called as your biogas. So we not only kill the bacteria, we also use them also to create biogas. To create biogas, students, to create biogas. So we get biogas from here. All the anaerobic bacteria will give the biogas. That is called as STP. Now, students, let me give a small recap. In filtration tank, we did something called as sequential filtration. Step by step, we removed all the thing. In sedimentation tank, because of gravity, whatever is present on the bottom is called as primary sludge. Primary sludge was sent back here, sent back here. Top part was sent to the aeration tank. In aeration tank, what did we do? We released oxygen to increase the aerobic bacteria. Once bacteria increased, once there was a increase in bacteria, they decreased the BOD eventually. BOD was decreased because all the organic matter was eaten by the bacteria here. Then in settling tank, we let all the bacteria settle down. Now some amount of activated bacteria is sent back here, that is called as activated sludge, and remaining is sent to the next tank here. The top part is sent to the water body. This anaerobic sludge digester, all the aerobic bacteria is killed here because of no oxygen. And this anaerobic bacteria here will give you biogas. Anaerobic bacteria will give you the biogas here. So that is the story of STP, students. Crazy story, right? That is the story of STP. STP, students, STP. This is what happens, students, in nature. Not nature, we'd make it happen. Students, clear, all of you? Clear? And students, did you know that less number of STPs, large quantity of unfiltered sewage water directly released into the natural body is been done. This is what happens in India. There are very less STPs, more waste removal because of population. What to do, students? So there was a situation that is near the lakes, Ganga Lake and Yamuna Lake. There was a Ganga Action Plan and Yamuna Action Plan. This Ganga Action Plan and Yamuna Action Plan, because of this, STPs were made here. Near these water bodies, we made what? Something called as STPs, right? STPs were made near them. Okay. So, build sewage treatment plant near the large number. No untreated sewage has to be done. This is called as SIG, Action Plan. Okay, students. Here is the question. Let's see who can solve. So, where do we remove suspended solids, students? Where do we remove suspended solids? All of you, the answer. Students, you tell the answer, I'll drink some water and come. Tell the answer, tell the answer. Adding some water. Okay. Which of the following sewage treatment removes suspended solid? That is going to be your, students, what? Suspended solid is what, students? Suspended solid. That is nothing but physical removal of waste, students. Tell me, where does it happen? Where do you think physical removal of waste happens? Primary treatment. Yes or no? All of you, primary treatment. Okay. Next question. A high level of BOD, biochemical oxygen demand, indicates if there is increased BOD is there. What is the meaning of increase in BOD, students? Water is pure. Water is highly polluted. Yes. Water is highly polluted. Water is highly polluted. That meaning is that. Okay. Okay. What is biochemical oxygen demand is a method for. What is the use of, what is BOD? Estimate the amount of organic matter present in sewage. Okay. Working out the efficiency of oil driven automobile engines. No. Measuring the activity of Saccharomyces. No. Working out the efficiency of RBCs. No. So A is the answer. So what is, students? Estimating the amount of organic matter in the sewage. More BOD, more sewage. Less BOD, less sewage. Okay, students. Next question, students. Okay, students, have you seen this in your native areas? Students, have you seen this in natives, in villages? Students, have you seen this in villages? There will be one Aji will be there. Next level accuracy. One paste from here, one disc throw, it will go and fly and go form here. Yes or no? She'll throw here, it will fly and go and stamp here. Yes or no? It goes stuck, gets stuck here. Now this is called as gobar gas making. Now why are they using cow dung? What is special in cow dung? How can we use cow dung to get energy? Now students, we will learn that now. Okay, now we will learn that. Okay, gobar, exactly. Gobar students. Students, have you seen this? So this is a obviously cow, right? Cow generally eats grasses. Grass has something called as cellulose, and this cellulose, when it goes to the stomach, that is rumen here, it has a bacteria in the stomach. This cow has a bacteria in the stomach called as methanogens. Now these methanogens will act on the cellulose and break them down. Students, see this, all of you. This cow dung and this methanogens, right? Methanobacterium is the example. Methanobacterium is the example of methanogen. This methanobacterium is present in the cow dung now. Now this cow dung can be taken because this cow dung has cellulose, little bit of cellulose here. It has the bacteria. So it has bacteria, it has source, cellulose is present. So cow dung has cellulose and it also has methanogen, that is methanobacterium. Okay. Now you take this cow dung. Okay. When this cow dung undergoes anaerobic respiration, when the bacteria undergoes bacteria along with the cow dung, when the bacteria undergoes anaerobic respiration, it will produce biogas. So let me just teach you one more time. See this. First we have the cow. We have the cow here. Cow will eat the grass. Students, grass has what? Grass has cellulose is present. Now, after eating, it will give cow dung. Cow dung we get. Now, students, this cow dung has what? This cow dung has two main components. It has the little of, it has cellulose, undigested cellulose. It also has bacteria. It also has the bacteria. Students, which bacteria? It has the methanogen. Methanobacterium, or also called as methanogen. It is present in the cow dung. Now, this bacteria, when it undergoes anaerobic respiration, this bacteria using the cellulose, it undergoes anaerobic respiration. Friends, what do we get? We get the biogas. Students. We get the biogas. See, students, biogas. What does biogas has? Biogas has hydrogen, it has methane, it has CO2. All of them are present part of your biogas. Hydrogen sulfide, it has hydrogen, methane and CO2. You can use for lighting, cooking and fuel also. Okay. Now, next question to all of you. Now, this is happening from the cow dung. But how can you make it in a very large scale? If you want to make it in a large scale, students, have you seen this in villages? Students, have you seen this in villages like this, something like this? These are called as what? Biogas plants. Inside this, we add the cow dung and we get the biogas. This is how it looks, students. So, biogas plant. In India, production of biogas developed was IARI and KVIC, the Indian Agriculture Research Institute and Khadi and Village Industrial Commission. Both of these bodies, they made the biogas very popular, students. This is a biogas. This is how the biogas looks like, students. Biogas plant looks like. Okay. Now, what do we do, students? Here we add the slurry. In the slurry tank, we add the slurry. Now, students, what is slurry? Slurry is nothing but cow dung plus water. Slurry is nothing but, students, cow dung plus water. We take cow dung plus water and we add it in the slurry. Okay. Now, once this slurry goes into this underground region called as digester. Once it goes to the digester, this is where the magic happens. Now, what is happening, students? In this digester, there is cellulosic material. From where? Cellulosic material from the cow dung. It has bacteria, that is methanogens, and here there is no oxygen here. So what do we get, students? What do we get? We get biogas here. So we have cellulosic material, we have the bacteria, so we end up getting the biogas here. So this bacteria under anoxygenic condition, in anoxygenic condition, there is no oxygen here, it will produce the biogas. So all the gas will come from here, and this is called as gas holder on top. Gas holder. Students, have you noticed when you try to boil the milk, if you keep a plate on top, will the plate go like this up and down? Will the plate go up and down? Have you seen it will hold the gas? So here there is a gas holder on top. Whenever enough of gas is made, it will go on top. Gas outlet here, outlet. Now remaining part will go to the remaining waste will go to the sludge tank. Now this can be used as a fertilizer eventually. So the main magic is happening in the digester. In the digester we had the cow dung, which has cellulose. It also has the methanobacterium, and when methanobacterium, in the lack of oxygen, it undergoes anaerobic respiration. Once it undergoes anaerobic respiration, the bacterium can produce biogas. Biogas is made inside, students. See this. Cow dung plus water, slurry tank, digester. In digester, biogas is made. It goes to the outlet. Remaining part is sent to the digester. In this digester, what is happening? That is cellulose. Cellulose is broken down to biogas. How? Because of anaerobic respiration. By whom? Methanogen. By methanogens. Methanogens. That is the concept. That is the concept here. That is the ultimate concept here, students. Ultimate concept here. So here is the question. Start the answer. Which of the following statement about methanobacterium is not correct? Not correct here. Okay, not correct, students. Methane. Okay. It can be used to produce biogas. Yes, it can be is a biogas. They are found in rumens of cattle and their excreta. Yes. They grow aerobically and break down cellulose, students. No. Why? They grow anaerobically. They do not require oxygen. They grow anaerobically always. They grow anaerobically. Okay. The what gas are produced in anaerobic sludge digester? What gases do we get in anaerobic sludge digester? We get methane and CO2, methane, hydrogen sulfide and CO2. See, what do we get? We get the entire methane, hydrogen sulfide, H2S and CO2, students. Do you remember this from here? From here, students. Not biogas. I am talking about here. Here we have, students, this is called as anaerobic sludge digester. Here we get methane, CO2 and hydrogen sulfide. Hydrogen sulfide here. Okay, we get hydrogen sulfide here. Okay, sulfide and CO2. Okay, next question, students. Select the correct statement from the following. Biogas is produced by activity of aerobic? No. Anaerobic bacteria. Methanobacterium is aerobic bacteria? No. It is anaerobic bacteria. Biogas is commonly called as gobar gas. Is pure methane? No. It is not pure methane. It has students. Biogas is pure methane? No. Biogas is not pure methane. We have methane, carbon dioxide, hydrogen in biogas. It is not pure methane. It is not pure methane. Okay. Not with there. Not with there. Activated sludge segment in settling tank of the sewage treatment plant is rich in source of aerobic bacteria. Yes. Activated sludge, students. Activated sludge is rich in the aerobic bacteria, and we send them back here. Here we see here. Go back here, students. Here activated sludge is rich in aerobic bacteria, which we send back here. Rich in aerobic bacteria. Rich in aerobic bacteria, students. Now, next question. The technology for biogas production was developed in India mainly due to the efforts of who were the two bodies who put all the efforts into making the biogas plant, students? All of you. Who can tell the answer? IARI, yes. KVIC, so both A and B? Both A and B, students. Both A and B. Both A and B, students. So with that, students, we are coming towards the end of the chapter right now. In the end of the chapter, it's very, very simple, students. You don't have to worry so much. All you need to do is listen a few things, write down other different organisms here, and your chapter is done. Okay, this was the main concept here. The main concept here, students, was what? Main concept here was STP and gobar gas, and it is over. Rest of the chapter is very easy. Next 15 minutes, chapter is over right now, students. Next 15 minutes, chapter is over. Okay. Now, microbes we learned in household, we learned the cheese, bread, and everything. Industrial, we learned production of bioactive compounds, production of different types of your alcohol, antibiotics, and everything. Sewage treatment, we learned sewage treatment. Now, the last thing, we'll also learn the biogas production. So, all this is done. The remaining part is how we can use in farming right now. Okay, how can we use microbes in farming? We can use them as a pesticide. We can use them as a biofertilizer. So, fertilizer and pesticide. Now, let's learn. Okay, let's see. Now, all of you know there is something called as agrochemicals. Agrochemicals are the normal pesticides. You might have seen, students, a farmer will go and keep on spraying. Students, have you seen? A farmer will have a tank on the back and he'll be spraying everywhere. Students, have you seen that? A farmer doing that. He'll have a tank behind his back. He'll wear a tank like a water gun. He'll keep spraying everywhere. Now, that is called as pesticide, which is very harmful to the plant, very harmful to us also. Right? It's very expensive also. Yield cannot be increased further with extensive varieties using conventional methods of agriculture. Agrochemicals cause environmental and health related problems. They can cause. So, what is the alternative here? Alternative is microbes. Use microbes here, students. That is your organic farming, students. Students, have you heard this word? Organic tomato, organic potato, organic carrot. Very costly there, students. Have you noticed that? Anything you want to sell expensive, you add organic in front of it. So, organic food are very expensive, you might have heard, because they are grown without any chemicals. Right? Organic farming, very expensive food. Organic. Okay. So, we can use microbes for biocontrol agent and as biofertilizer. Now, one by one, we'll learn both of them, and the chapter is over after this. Students. Okay, see this. Now, how can we use them as biocontrol agents? Microbes used in controlling plant disease and pests based on natural predator. Now, for example, there is a mosquito which is causing havoc. There is a mosquito in my farm which is eating all my plants, eating all the plants. Now, you introduce dragonfly. This dragonfly will go and kill the mosquito. Problem solved. Similarly, we can use the concept of prey and predator. If the prey is spoiling, let's say one deer is eating your plants, what do? If a deer, all of you know what is deer, right? Deer. If a deer is eating all the plants, you bring on tiger. Tiger will eat you also, but at least your plants are safe. No, the tiger will come, it'll eat the deer and the plants are safe. The same concept of prey and predator we are using for microbes also. There are certain microbes which can eat other items, other organisms to keep your plant safe. Like we don't have tiger here. Instead of tiger, we have microbes here. Okay. Now, do not cause pollution to our environment and do not eradicate pests, but keep them under manageable control. They will not eradicate, will keep them under control here. Okay, students. Now, all of you might have heard about BT cotton, students. You might have heard. There is a chapter called as biotechnology chapter. In biotechnology chapter, you might have heard about BT cotton. That is the same concept, students. Bacillus thuringiensis. Microbe. We are using microbe to control the pest. This microbe has something called as cry genes. Cry1AC, Cry2AC. This cry genes can control the population of caterpillar larvae. Right? It can control the crystalline protein BT toxin. Right? So they are used for cotton. Right? So this BT can control the population of your caterpillar larvae. We can use fungi, students. We can use fungi. That is your, where is it? Trichoderma. Trichoderma can be used. Trichoderma will help against plant root pathogens. Trichoderma, students. Trichoderma, plant pathogens. It'll kill. The trichoderma will kill everything. So we can use a fungus to kill the pathogens. Fungus to kill the pathogens. Okay, we can use virus also. There is something called as baculovirus, nucleopolyhedrovirus. This virus will not kill anything else. It will kill only very specific insect, specific arthropods. It will kill all the insects and arthropods, kill them. But this has no effect on vertebrates, non-vertebrates and plants. So it is become very specific, no, very good. No, you want to kill only one insect. Instead of spraying all the chemicals and killing everything, you introduce a virus. This virus will go and kill only the arthropods. Crazy, So we have a very specific solution here. Just because you are bad at chemistry, do you sit and study everything? Biology again, physics again? No. You will study only chemistry? No. So similarly, if there is a problem with the arthropods, you introduce this virus, baculovirus, it will only kill the arthropods. It will not touch any other thing. So best, students, it's best it is. See, economically approach to pest management involves multiple approaches. Baculovirus are desirable factor for IPM. What is IPM, students? What is IPM? Integrated pest management. Now, what is this, students? What is this integrated pest management? Understand. What is this integrated pest management? Understand, students. Knowledge of pest, their life cycle, their interaction with the environment helps in the developing a holistic approach in managing the pest. So we are not killing the pest, we are understanding the life cycle, we are observing them, and after observation we realize, wait a minute, this pest can be controlled if you introduce this one more virus here or one more microbe here. So microbe is helping us to control the pest. And baculovirus is the best because baculovirus will never kill the other organisms. It is very target specific. It is like a missile. This missile will only go and kill that arthropod. It will not kill anyone. Very amazing missile it is. Missile. Okay, missile, students. There is aphids will kill the ladybird will kill the aphids. Dragonfly will kill the mosquitoes. That is the, student. That is the entire biocontrol. So we can control the other organisms using microbes. Okay, students, using microbes. We have the examples, students. Understand, examples are here. These examples are given. Ladybird will kill the aphids. Dragonfly will kill the mosquito, but they are not microbes. Remember, nucleopolyhedrovirus is target specific. It will kill all the arthropods and insects. We have the trichoderma, which will kill all the plant root pathogens. And we have the BT, Bacillus thuringiensis. It will kill all the caterpillar larvae. So first one, second one, and third one. Remember these three. Now these two also you can remember, 4 and 5. These 5 examples you have to remember, students. 5 examples you have to remember. These 5 examples you have to remember. 5 examples of biocontrol, students. Biocontrol. Okay, you have to remember, students. Now, how can we use microbes for biofertilizer, now, students? Fertilizer. Like we use them to control the pest. How can we use them to control the? How can we use them as a fertilizer now? Okay, fertilizer. Students, have you seen this? Students, have you ever noticed this? It is present in your leguminous plant. It is present in your leguminous plants. Okay. So in the case of leguminous plants, right, in the roots there is something called a root nodule formation. In that root nodule formation, we have a organism called as rhizobium. And this rhizobium will help in the nitrogen fixation. It will help in nitrogen fixation. It's a free living acetobacter. It is free living. Right, sorry. This is a rhizobium in symbiotic. Rhizobium is symbiotic, help in nitrogen fixation. Okay, that is your rhizobium, students. Rhizobium. Okay, rhizobium. It is in symbiotic, while the acetobacter is free living, students. Acetobacter and also acetobacter. They're free living. They do not found symbiotic. Students, see here. Acetobacter, rhizobium, both will the atmospheric nitrogen and ornithopod, the nitrogen fixation. See, this is the roots. Then there is mycorrhiza also. Mycorrhiza is a symbiotic again. Mycorrhiza from this from the glomus, they help in absorption of phosphorus. So mycorrhiza can help in absorption of phosphorus from glomus. Okay, genus glomus. Okay. Students, here we can also write Azotobacter and Azospirillum. Okay. We also have the Azospirillum. This also is free living. Free living. Then we have cyanobacteria also. Cyanobacteria can be used as a biofertilizer. Anabina, nostoc, both can help in hydrogen fixation. Anabina, nostoc, even oscillataria, but oscillataria does not have the heterocyst. Does not have the heterocyst. See, it can be added in the paddy fields or the paddy fields. Okay, they will add organic matter. For students, they are also commercially available like this. So students, here is a mind map. Use this mind map to solve all the questions, students. To solve all the questions. Okay, so use the mind map to solve all the questions, and you will be able to get better marks. Okay, so students, I have few questions here. Let's try to solve those questions. I have a few questions for all of you, students. I have a few questions for all of you. Students, let me the answer. All of you. We'll do rapid fire, students. We'll do rapid fire. 10 minutes, 15 minutes, we'll wrap up the entire thing now. Okay. Which of the following microorganism used for production of citric acid? Aspergillus niger. All of you should know, students. All of you should know. Aspergillus niger. Biogas consists of, students. Biogas has what? Biogas. Biogas. Carbon dioxide, methane, and hydrogen. Methane, hydrogen. Okay. Which of the following microorganisms used for Swiss cheese? Swiss cheese, students. Swiss cheese. Propionibacterium and Shermani. Propionibacterium, Shermani. Next question. A modern farmer can increase the yield of paddy up to 50% by using what, students? Paddy. I told you just now. Paddy is a rice. We use cyanobacteria. Cyanobacteria. Which of the following is wrongly matched of the microbe and industrial product? See, Aspergillus niger citric acid. Yeast is statins. Yes. Yeast, students. Statins only. Acetobacter aceti. No. Lactic acid is LAB. Students, statins is from yeast only. Okay. So remember that. Okay, students. A drug used for dash patients is obtained from a species of organism dash. Okay. AIDS obtained from Pseudomonas, no. Drug used for heart is from penicillium, no. Okay. No. A drug used for organ transplant to make sure there is no graft rejection, we use cyclosporin A. Cyclosporin A is obtained from Trichoderma polysporum. Trichoderma polysporum. Good question. A patient brought to hospital with myocardial infarction is normally immediately given. They give, students, they give streptokinase to make sure there is no clot bursting. Clot bursting, students. Now, the most abundant prokaryote useful to humans in making curd from milk, that is lactose. That is your lactose. As a bacteria, heterotrophic is a, talking about LAB only here. LAB is a heterotrophic bacteria. Okay. Molasses purpurus is a yeast used commercially, students. Molasses purpurus, just now you learned. It is a yeast used to blood cholesterol lowering agent. Blood cholesterol lowering agent. Okay. Here. Blood cholesterol lowering agent. Next question, students. A river with an inflow of domestic sewage is rich in organic waste may result in. If there is more organic waste, students, these are difficult questions. A river with an inflow of domestic sewage rich in organic waste may result in drying of the river very soon, algal bloom, increased population of aquatic food web, increased production of fish due to, students. Why? Because if there is an inflow of sewage, more organic matter, more bacteria, more O2 demand, If there is more O2 demand, less O2 in water. If there is less O2 in water, death of fish due to lack of oxygen. Because of lack of oxygen, death of fish. Crazy question, students. See, BOD may not be good indicator of pollution, indicator of pollution for water bodies receiving effluent, students, from effluents from, may not be a good indicator. BOD is not a good indicator, students. Petroleum industry, students. Why? Petroleum, because in petroleum we do not get organic waste. In sugar, domestic dairy, we get organic waste. Organic waste can be shown by BOD, but petroleum waste cannot be shown by BOD. Okay. So in these questions we have done already. Immunosuppressive agent, Trichoderma polysporum. These questions are made you do in the class itself. These questions are made you do in the, they are made you try in the class, students, class itself. In the class itself. This we did today itself. Right? This we did today. All these questions we did today itself. Okay, organism uses biofertilizer for raising soybean crop. Soybean, students, is a legume. Soybean is a legume. If it is a legume, rhizobium. Legume, rhizobium. Okay. See, rhizobium. Leguminous plant. Fuse rhizobium. Students, here is your, I have a homework question for you, students. This is your mind map. This is your mind map. I have a homework question for you, students. Homework question is very simple. I want all of you to tell me the names of, or wait. Homework question. In comments, students. Homework question in comments. This is the question for all of you, students. Now, students, again, I'm telling you multiple reminders. DNA batch has already started, right? Students, those of you are asking me, sir, how do I join DNA batch? Students, if you look at the description, there is a link there for DNA batch. You can click on that link and you can join the DNA batch. It is for, price has been reduced, so please go check the batch. If you want Tattva book, there is a combination that is the best combination, because Tattva book you'll get a hard copy of it. That's the best combination, students. In DNA batch right now I am teaching anatomy of flowering plants. Next class I'll be starting the dicot stem, so please join as soon as possible, and I'll see you in the class, students. Until then, take care all of you, and bye all of you, students. Bye.